So we'll look into microRNA. So without delay, we'll start the class. Okay, so first basics. First, we have a cell, right? In that cell, we have nucleus, cytoplasm. Cytoplasm is the jelly in the cell, okay, the medium of the cell. And there are different organelles, and the prominent organelle which you can see here is these. These are called, is it visible? Yeah, these are called ribosomes. What are ribosomes? Wait, what are ribosomes? Factories of protein, okay? Ribosomes are factories of protein. So where proteins are formed are called ribosomes. So first, this is nucleus. Nucleus has genetic material. Then ribosomes are the places where proteins are formed. So nucleus has to tell these ribosomes in a way so that they can form proteins. What is a protein? Anything, any hormone in your body is a protein, okay? So firstly, in this nucleus you'll have DNA. Firstly, before DNA, I mean DNA to definitely you'll have. So in nucleus you'll see these are chromosomes, okay? How many chromosomes do we have? Jamuna, right? Yeah, how many chromosomes do we have? We have 23 pairs of chromosomes, of which 22 are homologous. That means not, quite same. And then the 23rd chromosome is sex chromosome. XY for male, XX for female, okay? We have 23 pairs of chromosomes, and this is a chromosome, okay? In chromosome also, although I didn't discuss in the class, we have two chromatids like this, okay? For now, forget about chromatids. This is a chromosome, okay? What is chromosome? It is a tightly wound DNA. That means if this is a histone, just know the name. If this is a histone, onto this histone, I'll tightly wound my DNA and I'll make it a huge structure. That is chromosome. So which one is bigger, chromosome or DNA? You can't really say that because chromosome is made of DNA, okay? When I ask you which one is big, gene or a chromosome? No. When I ask you if gene is big or chromosome is big, I'll tell you what is a gene in a while, okay? For now, just focus on this. This is a chromosome. Inside chromosome, if you open up the chromosome properly, you'll see that, it is wound around these small yellow particles, right? These are called histones. Now, if you just look at here, this is a double helical structure, double helical and double stranded. You have two strands of chromosomes, okay? And if you open it up a little bit, you'll see ATGC or ATCG. But remember ATGC, okay? ATGC. What are these? These are called nitrogenous bases, okay? These are called nitrogenous bases on DNA. I will tell you the difference between DNA and RNA, because in RNA we'll look at, ma'am, in RNA we'll look at, no, AUGC, okay? AUGC. There is no thiamine in RNA. We will look at this, although we've covered in our previous classes. I just wanted to tell you that DNA is a double-stranded, double helical structure, okay? But RNA is a single-stranded structure. Although there can be a double-stranded RNA, but RNA in general is a single-stranded structure. It has AUGC, DNA has ATGC, okay? I want you to know this, I want you to remember all of this, okay? Now, moving on. See, genetic material. Firstly, we've seen the basic difference, right? If someone asks you, what is a chromosome? You must be telling them ki chromosome is nothing but the DNA wound together on histosomes closely packed. Okay, that is chromosome. Then we have genetic material. What is this genetic material? Anything in your body or a virus's body, which codes for the basic form of life, right? So genetic material can be DNA, can also be RNA. It can be DNA and it can also be RNA. So DNA is more stable. I'll tell you why is it more stable, because it's doubly stranded, right? You can't take away something from it. And RNA is relatively less stable. No, you cannot say RNA is less stable, but RNA is relatively less stable. Now what do I mean by more stable and what do I mean by less stable? What do I mean? I mean that if I say something is less stable, then I mean that the rate of mutation in that particle is relatively higher. For example, COVID, coronavirus. What happens with coronavirus? It mutates now and then, and it's creating struggles for us. You know, there was an alpha, beta, gamma variant, right? So corona was an RNA virus. So when I say something is less stable, then that means that the mutation rate is higher in that particular genetic material. Okay? Yeah. Now quickly tell me, what is the full form of DNA? Come on. DNA. Deoxyribonucleic acid, right? Yeah. RNA? Ribonucleic acid, that's it. Yeah? Yeah. So, I want you to know this, and there are different types of RNAs. We will now discuss about all these types of RNAs. Firstly, messenger RNA. Okay, I'll tell you what exactly it is. Just know the name, messenger RNA. And then there is microRNA. This microRNA, I will also write it as, ouch. Yeah, this microRNA, I will also write it as, miRNA, okay. First there is messenger RNA, then there is microRNA, then there is transfer RNA, and then there is ribosomal RNA. Okay, messenger, micro, transfer, ribosomal. Clear? Any doubts until there? Because after this point it's gonna get a little complex. Okay, so now we'll look at the glossaries. First, glossary may so you don't have to make notes because everything is in here. Ribosomes are factories of proteins. That means the proteins are made in ribosomes. All right. And how many ribosomes do you have? Many. Okay, you can't count. Many. Simple. But how many nucleases do you have? Only one. Only one. Okay. Chalo. And nucleus is in which cell? So where do you find nucleus? Only in eukaryotic cells. In prokaryotic cells such as bacteria, you won't find a nucleus. You'll rather find a nucleolus. Alright. So true nucleus is always found in eukaryotic cells. Fine. So first, ribosomes, factories of proteins. Cool. Next, there is this process called the transformation of DNA into RNA. There is this process called transformation of DNA into RNA. That is called transcription. Okay. DNA to RNA is called transcription. The process of RNA getting transformed into protein is called translation. Now I told you that everything we have in each of our cells is in our genetic material called DNA. Now this DNA has to tell our ribosomes to make a particular protein. For instance, let's say there is this, you got some cut. Now there has to be a clotting factor which has to be made, right? Although blood has clotting factor, but who will make this clotting factor in the first place? So DNA in your red blood cells will ask, not red blood cells, yeah, DNA in your stem cells in bone marrow will ask your, what do you call that particular stem cell or that particular other cells to make something called as clotting factor. Okay. Now how does the procedure happen? First DNA will, you saw the image, right? In this image, you see this is nucleus, DNA is here, ribosomes are floating around in cytoplasm. Now someone has to tell these people that you have a message from here, right? You got a message from here. So first what will happen is DNA will convert itself into an RNA, especially messenger RNA, and then the message will be passed to ribosomes. But only if you get the message can you make a protein. No. Why? You need ingredients also. I'll tell you recipe, you can't make biryani, right? You have to need ingredients. Now who will get you these ingredients? There's something called as transfer RNA. And where does that transfer RNA come from? Also from DNA. So DNA tells this is the message I want you to make, And DNA will also tell these are the ingredients you need to make this. So again transfer RNA will also come. Okay. Now once you have the message recipe, then all the ingredients, now can you actually make it? You need someone to make it also. Ribosomes will make. But what is inside ribosomes? There's something called as ribosomal RNA which will actually make the protein. So first I told you messenger RNA, what does it do? Messenger RNA will take a message from DNA to ribosome. This process is called transcription, right? Transcription, DNA to RNA, transcription. Next, tRNA, that is transfer RNA, will get you ingredients to make biryani, right? This biryani is protein. Now, then actually who will make the biryani? It is ribosomal RNA or rRNA, okay? I'll tell you what is that. But, so this process, translation. So if you are finding any difficulties to remember, first a book, a transcript is made. Then later you will translate to different languages, alright? So this is the process I want you to know, and then this is the procedure of making proteins. First from DNA you will have messenger RNA, transfer RNA and rRNA, right? So this is recipe, okay, recipe spelling wrong anyway. So is it right? No, right no. So ingredients, chef, okay. So far fine. Now we'll look at something called as gene. Something called as gene. Gene is simply nothing but coding part of your DNA. Now your DNA is very long. Your DNA is very, very long. In that particular DNA, you'll have parts of, areas. I'll have to draw and show. So, let's say this is your DNA. Now, how do I represent DNA? Although technically, if I have to represent, I have to draw like this first. There is something, and I have to draw something like this. Yeah. Then I have to draw something. In here, A, T, G, C, A, like that. It can be T, T, G, G, C, 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 something like that. And this particular thing is called a sugar phosphate backbone. This is always the same. This is always the same. There won't be any change. This is on sugar, you will have this nitrogenous base. On sugar, you will have this nitrogenous base. And there is phosphate attaching to sugar. Okay. So it's this particular part is called sugar phosphate backbone. Okay. This is sugar phosphate backbone. Okay. And what is the sugar here? What is the sugar here in DNA? Deoxyribose is the sugar. In RNA, simply ribose is the sugar. And in RNA, you won't see this bottom part. You won't see this bottom part in RNA, okay? You'll only see the top part in the RNA. So, if I have to actually write about DNA, you see this is a sugar phosphate backbone. To be very clear, see this particular part I am talking about is a sugar phosphate backbone, okay? On that you will find nitrogenous bases. And also, is this the right representation? No, because it is like this. Imagine this ladder, I am twisting it like this. It's going to be like this only. For the purpose of being more clear, I have just opened it up. Okay. So, this is sugar phosphate backbone and you will have different nitrogenous bases on top of sugar. Of course, on top of sugar phosphate backbone, but where does this nitrogenous base actually reside on? On sugar. On sugar, okay. Yeah. Where is this? Yeah. This is something I drew, right? This is sugar phosphate backbone. I'll draw like this. Sugar. I won't draw phosphate, okay. Sugar again. Sugar. Sugar. Now you tell me what is supposed to come here? Here. T. Okay. Good. Ikkada. You forgot? Do you remember? U is for RNA. G is for DNA. Why? Because A always pairs with T and G pairs with C. That's why I asked you to remember ATGC. Okay. A always pairs with T G always pairs with C. Okay. So what does it, what does come here? C, right. Here G, here T, right? Yeah. Now, for the purposes of simplification of this class, can I simply write, if I have to write about DNA, I can simply write A T T T G C C G A, right? It can either be this part or it can either be this part. It doesn't matter because these people will have the complementaries which we already know, right? So, for all the practical purposes, I can simply denote our DNA to be ATGC, 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 right? Yeah. Now we are looking at genes. Now, see, this is ATGC, DNA, this is phosphate, sugar, nitrogenous base, right? And this particular setup is called, this part is called, this is called, pardon? Yeah, diagrammatic representation. No, it's called a nucleotide. You guys have to revise, okay? Because you were not, I mean, you didn't see the videos, right? But both of you were regular. Yeah, nucleotide. See, biotechnology will account for, if there are 15 questions from SNT, biotechnology generally accounts for 4 to 5 questions every single year, okay? You cannot skip biotechnology and go. Nucleotide, okay? This is called a nucleotide. Fine. Now, what are the nitrogenous bases? Again, you don't have to remember the names, but I'm just telling adenine, thymine, guanine, cytosine. Now, in RNA, you'll find AUGC. I mean, that means U is for uracil. You will not find thymine. You will not find thymine, okay? Here, yeah, if I show you, this is a DNA setup. RNA is simply like this. RNA is only single stranded. I told you RNA is single stranded. All right. Now single stranded. Now you see, how does this mechanism of transcription work? A very simple thing. Mechanism of transcription works something like this. In my DNA, firstly I have something like this, right? Now I have to open this up first. I have to open this up. That process is often called as denaturation, but you don't have to remember the name of the process, but just know DNA is getting opened. So it's now open. Now any particular part, you take any particular part, let's say A, G, C, C, T, A, G, G, T, T. Yeah. If this is your DNA, if this is your, can everyone see it? Yeah. If this is your DNA, now tell me what will be your RNA? What will be your RNA? First, U here, because A codes with U, there is no T in RNA, right? AU, then G, C, here, G, A, this T, A, next, U, right? This is your RNA. For the purposes, we'll call it messenger RNA. Okay. So first from your DNA, we made something called as RNA. Yeah, any doubts, ma'am? You sure? Yeah. DNA, RNA. Now from this mRNA, you will take it to ribosomes, right? Now the thing which you have to keep in your mind is that this particular part is a single strand. So how, I told you DNA itself has to open first. Why? To make a complementary RNA. Right. Now this will float and go like this. Only when, if this is a single strand, it can transform itself into protein. I repeat this. Only when this mRNA is a single strand system, can it be translated into protein. Ante, if this is not single stranded, if this is double stranded, can you make it into protein? No. Why? Because if I add a complementary element here, then I have to open this up first, right? And for RNA, opening up is hard. Why? It's not stable. The moment you try to open, it will disintegrate, it will digest. Okay. So I'm telling you that only a single stranded RNA, I repeat, I'm telling you only a single stranded RNA can translate into a protein. Okay, only a single stranded RNA can translate into a protein. Why? Because only when you have open end, then other parts can take your material from here. This is A, it will take something, amino acid or something, okay. So I repeat this again, only when your RNA is a single stranded RNA can you actually make it into a protein, okay. Yeah, fine. Stroke eraser. Fine. So see, if this particular part is made a double strand, now how can you make this a double strand? How can you make this a double strand? Simply add something which is not here. Here T will come. No, U will come. Here A will come. Okay. So I'll write it again. Here U, here A. Why? These are complementary. No, this is RNA. AU again UA, right? So if you make this double strand, can this RNA transform into a protein? No. So if made double stranded, it can't make a protein. Okay. Yeah. Now these are all the basics I want you to know, and yeah, there is this illustration also which you don't have to worry about. AT, GCC, T, all that changes here. Okay, AU, TK, A. All right. So this is something which we've discussed. Now these are all the basics. Now let's move into the actual subject. And before we move into actual subject, I want to discuss 2006 Nobel Prize. Now why am I discussing 2006 Nobel Prize? Right. There is some method to this madness because do you know the book list of SNT? What you have to read for in general? They say NCRTs, right? But you can't read NCRTs for SNT. I mean, although you can technically, but I would say in the beginning, if you start with NCRTs, it will just be a futile effort because either you might have known something from school or you'll end up reading a lot of material which is not actually relevant. So rather you would, I would suggest you to pick some standard material or some coaching notes and start. However, there's one material which is non-negotiable and you have to read it. That is your 12th class biology textbook. 12th class biology textbook mein, not all the chapters have to be read. However, I would suggest you read all the chapters, make notes from all the chapters. But in particular, biotechnology, chapter 9, chapter 10, chapter 11, somewhere around that. Okay. So you have to read that. Now in that chapter, you have something called as RNA interference technology. Now again, you might ask, sir, question yes, question did not surface on RNA interference technology as such. However, in this very particular chapter, if you look at it, you will have BT cotton as a topic. Just below BT cotton, you have RNA interference technology. So BT cotton was asked many times. BT cotton, at least I can count 3 times it was asked, BT cotton. Okay. So RNA interference technology is the next important thing, and since there is already a Nobel Prize on microRNA, this also gets importance. Alright. So now the focus is on RNA interference. Now see, simply in SNT and economy, the names are pretty simple. You don't really have to worry about what does it mean. Simply you are interfering RNA. You are interfering RNA. If I have to put it in a very small nutshell, I will say that RNA interference technology is making this messenger RNA double stranded. is making this messenger RNA double stranded. How do I do it? I'll tell you. But what happens if I do it is something you have to know. What happens if I do it? If I make my messenger RNA double stranded, what happens? It doesn't change into a protein, right? It doesn't change into a protein. Yeah. Now we'll look at something which is interesting. So firstly, if there is a, how do viruses work? How do bacteria or any kind of pathogen work in your body? Firstly, let's say a virus entered your body. Let's take this as this is adenovirus, okay? And this is COVID. This is the RNA inside. RNA inside, okay? And this is this on surface. What do you have? On surface of coronavirus, what do you have? Spike protein. On the surface of coronavirus, you have something called as spike protein, okay? We will discuss this when we are discussing vaccines in detail. But for now, you have to know this basic part. Ki on surface of corona, you have spike protein. This is an adenovirus. What do I mean by adenovirus? Okay, common cold virus, okay? Common cold virus is adenovirus. Now, this also has some genetic material, right? This also has some genetic material here. Now, how do these viruses work? In general, if a virus enters your cell, any of the body cell, how does coronavirus enter? It enters using the spike protein, and there is something called on our cells. On our cells, see, every cell has some proteins on its surface. Every cell has some proteins on its surface, okay? Now, this coronavirus enters your cell using some receptors on our cells. Those receptors are called ACE2, or doctors will write it like this, ACE2 protein, ACE2 receptor, okay? Now, why did I say doctors will write like this? Because in 2021 prelims, question came like this, ACE2 protein karke. What is ACE2 protein? Protein, they didn't even ask what is ACE2 protein. They simply asked the term ACE2 seen in the news recently refers to, there was this antigen on virus karke. The only one option of biology was that part, and it's called antigen. I'll tell you in health class. So one of my doctor friends, she is from Usmania only, and she thought ACE2, no, something is related to IT. They write it like this, ACE2. So she put something else and came. She didn't clear that year. So these things have to be kept in mind. Keep ACE2 is different. I mean, like people write it like this, but in newspapers etc. they wrote it like this. So whenever UPSC goes with an answer, no, it takes the most simplest form. This is the most simplest answer, okay? So just know that. So on our body cells, on our body cells, you have something called as ACE2 protein, and this virus, coronavirus, enters through spike protein, and through this ACE2 protein, it will come inside your cell. Now, even if you don't understand this point, you will understand in the vaccines class. Don't worry. But now, what happens when actually virus enters? In fact, not just virus, any bacteria enters your cell. However, in your cell, if there is a pathogen, how does it react? What does this pathogen try to do? In general, bacteria same just say. So the bacteria will fight against you, huh? No, what does bacteria do? Yes, it will replicate. Any particular organism, it has to live, it has to reproduce, it will replicate itself. Now, how will it replicate? This is the question. So, for virus, I will not talk about bacteria today, but for virus, how will a virus replicate? Virus lopala, there is no ribosome. Did I draw any ribosome? No. If you look at that adenovirus also, there is no ribosome again. So, ribosome lekunda, can it make proteins for itself? No. No, right? And I told you, even RNA itself is a compound which can be created, right? It also can be created by ribosomes. So this virus will use, I repeat very importantly, this particular virus or pathogen will use our own body's ribosomes. This virus will use our own body's ribosomes. What are ribosomes? Factories of protein, to make the proteins for itself. Ante, there is our nucleus. This is our nucleus. This has some DNA. From this DNA, we will make our RNA. What is this RNA? Messenger RNA. But our virus here is also having some RNA. Now, what does the smart virus do is, it directly sends this RNA to ribosomes. These are ribosomes. Okay. Now, this will take time. No, it has to first transfer and come. Directly, this virus goes to ribosome, and it will now make. Let's say this part is RNA virus. No, okay, AUUGCC. Right. Now it will replicate this. This particular code is directly given to ribosome. Some protein is formed. So now just remember one part. Viruses give their RNA or their genetic material directly to our ribosomes. Okay. So our ribosomes will use our energy to make viruses. And what is this extra virus? This protein is made. This protein will become into an extra virus. When I discuss about virus, I'll simply tell you virus is nothing but a genetic material. Okay. So this extra virus will now go and attack your other cell. Right. Slowly what happens? Your body will have more and more viruses, and since your proteins are not being formed, your healthy functioning will affect. Why? Because your ribosomes don't have infinite energy, right, to make everything. You have limited energy, and that particular energy is also being used to make copies of viruses. No, that particular energy, ribosome has some energy, and how does energy come? Mitochondria. Okay, so that particular energy is used to make copies of viruses. So only one thing which you have to know is that viruses, when enter your body, they replicate. They replicate using our own body's mechanism, thereby depriving of our own resources to us. Okay. Yeah. Having said that, now, yeah. What if? So I told you this is for virus. I'll tell you same thing happens in tobacco plant. Tobacco, or I'm not sure, but in some plant it will happen. But here, this is not a plant virus. There is something called as a nematode. Okay, nematodes are nothing but worms. Okay, when I talk to you about nematodes, worms, these are very important for two reasons. First is, all of this research of microRNA was done on a worm called C. angulis or something. Spelling might be wrong, just go look at it later. But all of this, why have they chosen small worm like this, small nematode like this? Why? Because almost all of these C. angulis worms have roughly 1000 cells only, just 1000 cells, and that too, in that 1000 cells, that is 1 millimeter length worm will have differentiated cells. Now, what do I mean by differentiated cells? It has a gut, it has a stomach, it has different cells like hair. So it has differentiated cells. So just 1 millimeter long worm has different cells, and you can, when you actually count every single cell, you can go to 1000. And all of your RNA research was done on this worm. Okay, was done on this worm. Now, this is the first reason why the worm is important. Second reason is that these worms can be pests. These worms can enter your plant. Okay, there is your plant. If these worms enter your plant roots, will have these big nodules. Okay, this is not healthy for plant. I'll tell you why. Because when the worm enters your plant via roots, what does it do? It releases its own mRNA. This is a worm, it has its own DNA. It will quickly open this DNA, transcript into mRNA. It will quickly open its DNA, transcript into mRNA, and it will send this mRNA to plant. Why? Because it itself doesn't have energy enough to grow a protein of itself. So now plant has a lot of energy, right? From photosynthesis, energy is coming. Now plant will think, plant's ribosomes will think, okay, some mRNA came, now I have to make a protein. It doesn't know, okay, this is the wrong recipe, I should not make it. It will make. So now the plant is affected, worm is growing. Plant is affected, worm is growing. So what will happen? Loss. Because although the plant might look from the top, key it is good, you won't see it flowering. You won't see the grains coming a lot. Things like that. Okay. So the point here is, whenever a nematode gets into a plant root, it uses plant's own system to make copies of itself. Okay. Yeah. Now, if I want to stop the nematode, now is it practically possible for me to go into soil and take every single worm away? In a field, in lab also it's hard. In field it's even harder, and it's not practical also. So what scientists have devised that, they thought, let's make a transgenic plant. What is this transgenic plant? What is this transgenic plant? Oops, transgenic plant. What is trans? You might have heard cisgender, transgender, right? What is a transgender? Who is called transgender person? Huh? No. Why do we use different words then? No. That is super female syndrome. No. Okay. You're not aware of this basic factor? No. See, in simple terms, if I have to say, I have to describe a transgender person, transgender is someone whose biologically assigned sex is not something which is same in his mind or her mind. Okay, so his biologically assigned sex is different from what he thinks of himself. For example, a person can be a boy biologically, and he might like to be like a woman, like to, like women toys or toys, etc. And like to, dress up like a woman, thinks like a woman, doesn't have any interest in this boy conversations, things like that. So, in general, a trans person is a person who at the time of assigning his birth has a different gender than his mind. For example, it can be with a woman also. A woman can always feel like a guy, right? That's called transgender, alright. So, bisexual is when a person has sexual orientation towards two genders. That means he or she gets aroused and wants to have a, wants to have a relation with both of them. And sexually itself. Okay, that is a different word altogether. So my point is, whenever you see something called trans, you have to think different. It is different. Now when I say transgenic, genic is genes. Now when I say transgenic plant, that means this is a normal plant. Right. Now there is this normal genome. Let's say this is a normal genome of the plant. Now if I add an extra gene, which is not, if I add an extra gene which is not usually in this plant, in nature it is not found. I am adding an extra gene into the genetic material of this plant. How will I do it? I'll do it when it is in form of seed. When it is in form of seed, I'll simply try to add a extra small gene. Okay. So if I make a plant which is transgenic, that means having a gene which it doesn't usually have in nature. Right. So I'll make a new plant now. So yeah, this is my, okay, this is a bad color. Okay. Now this is my plant with some extra genes here. Okay, extra genes. Now I have deliberately inserted this particular extra gene to tackle nematode. Why? I know in that particular area you will find, different nematodes, that is worms. I put two genes for two famous nematodes, two different genes for two famous nematodes. Now how did I design these things? I told you that nematode, when it comes to a plant, it gives its own mRNA to ribosomes. It gives its own mRNA. This is worms or nematodes mRNA which is presented to our plant's ribosomes, right? Now, if I insert, let's say this is ATGC, very simple ATGC. If I insert TACG here, if I insert TACG here, this is in our own plant's nucleus. This is in our own plant's nucleus. This will transcript into, I'll simply add ATGC only here. I'll add the exact same thing. This is mRNA of nematode. This particular, okay, did you understand? Should I repeat again? Okay, I'll just repeat again. Listen. So, this is mRNA of nematode. What is nematode? Parasite or worm. What is parasite? Simply it is using your resources to live in it. Okay. Now, what will our body think? Our body will think this mRNA is of our own bodies, or plant will think this mRNA is my mRNA only. I will make a protein for this. If this is ATGCC, plant will make protein from this. Now, I have added extra, very same thing to my plant. My new plant has ATGCC already in it. My new plant is transgenic plant. I am adding an extra gene to the plant. Why am I doing it? I'll tell you now. That is called why is it called transgenic? Because I am adding an extra gene, right? Now this particular gene will now translate into AU, no? AU. T won't be there in RNA. AU. This will become UACGG, right? So now my plant has ATGCC in it. My plant will transcript this DNA into mRNA. My plant will transcript this DNA into mRNA. This mRNA will now go, it has AUGCC, it is UACGG, it will simply stick. Because these are complementary pairs, it will simply stick. Now, this single stranded mRNA became double stranded RNA. This single stranded became, now what does it mean? That's it. It cannot get translated. Why? Only single stranded mRNA can produce proteins, right? So this particular part is important. So it becomes double stranded. Now, this is called RNA interference technology, or few people might call it gene silencing. Gene silencing, okay. So this is how you do it. Jamna, any doubts? All cool? Okay, this particular single strand RNA became double strand RNA, okay, and thereby it cannot become a protein. Now what will happen if protein is not produced? This nematode will struggle to survive because its protein is not being provided. So this nematode will die inside, okay. Yeah. So see, application RNA interference technology is pest resistance against nematode for tobacco plants, and they call this the complementary RNA which we are making using what? Transgenic plant. This complementary RNA to the mRNA of nematode as siRNA, small interference RNA. They call it small interference RNA, and it is doubly stranded, right? Anyway, see, again, don't get into technical details because you don't have to become a biotechnologist. You have to just clear the exam. All right. So how does UPSC ask? I told you in the first class only, UPSC only focuses on what is the application of this particular technology. Next, will this be, for example, they asked 3-parent baby. So they said the procedure of 3-parent baby, can it happen before fertilization and after fertilization? Now this also has to be known, because the moment you do fertilization, there is already a baby here in your body. Although it doesn't have any rights, but there was a debate about the abortion rights, right? So that's why it's important to know that a particular procedure can be done before fertilization or after fertilization. So these are a few important topics where you have to know key how does this work is important, but what is more important is for what it is used. If UPSC were to ask a question, it will simply ask RNA interference technology recently seen in news is used for pest resistant plants. That will be the answer, okay. So application, pest resistance, and then working. Nematode needs to make its food using plants' resources for it to survive. If nematode's mRNA is interfered, then it can't survive. Here, obviously, it needs its genes. Yeah, I didn't tell you what is a gene, right? Yeah. So, you have, okay, definition of gene. I told some 1,000 times in the class. Gene, definition of gene. What? If you have to write in an answer, what will you write? You can't explain this way, right? A gene is a functional or coding part of DNA. Okay. A gene is a functional or coding part of DNA. Now, what do I mean by that? DNA can be very big. I mean, it is very big. So, in this, usually, not everything is coding part. For example, in DNA, you have only 2 to 3% of the genes. That means if there are 100 strands like this, I mean, if there is, I told you, if this is whole ground, big ground, only 2 to 3% part is constructed. Only 2 to 3% part is constructed. Now, what about the other part? They usually call it junk DNA. Although we don't know what exactly the purpose of that DNA is, which we will discuss in this Nobel Prize. But usually this part is called junk DNA. Which part? Other than genes. Other than genes. Okay, I'll tell you what is a gene first. So gene is this particular part. Gene is this particular part which usually gets translated into a protein. Now, will this part get translated into protein? No, it won't. It won't. This part of DNA will not get translated into a protein. This part also won't. But if there is a gene here, this part will. So if I have to tell you, gene is an area or is a sequence of your nitrogenous pairs, sequence of your nitrogenous bases. Okay. So sequence of DNA which gets translated into a protein is called gene. I repeat, sequence of DNA which gets translated into a protein is called gene. So there are areas, 97% of your DNA doesn't get translated into a protein. 97% of your DNA doesn't get translated into a protein. Okay, that doesn't mean it is not useful. I simply told it doesn't get translated into a protein. It might play a different role. We'll discuss about it. But can I simply say, and now, gene is always specific to a trait. I repeat, gene is always specific to a trait. Now, what do I mean by that? I mean, if I have a particular gene that is related to an eye color. So, if I have that gene, I'll have blue eyes. If I don't have that gene, I won't have blue eyes. As simple as that. But that gene will be called eye color gene. Now, there is a difference in this. I'll tell you, listen very carefully. If there is a particular gene, I call it eye color gene. That gene, let's say, this is an eye color gene, okay. If I have AGGCTTT, this will be your blue eye. Now, if there is a small change here, this becomes A here, T, same thing, same repeats, but let's say, okay, there is a different gene altogether, and there is a small change here. This becomes G, let's say. Small change. Your eyes won't be blue, they'll be black again. Okay. If there's a small, if there's a different gene here, it will be red eyes, something like that, yellow, green, whatever. My point is, whenever I talk about gene, I talk about the region in DNA. That means, if this is a DNA, I can tell you address of that gene. Let's say this particular area. Okay, SR Nagar, second floor, Hyderabad IAS, something like that. If I talk, and if that particular, I'm talking about address. In gene gurinchi maatladinappudu, I'm only referring to address. Now, why am I repeating this so often is because only in that part will you find a gene related to your eye color. That can be red eye, blue eye, green eye. Those are called alleles. Alleles are simply nothing but different forms of genes. Okay, spelling is wrong. Alleles. Okay, So My point is, whenever I'm talking about gene, I'm talking about set of nucleotides together at a particular place. At a particular place, and in every human being, if you go look into that place, you'll find a gene of that particular part. So whenever I talk about gene, I talk about I don't talk about eye color in specific. I talk about eye color in general. If I say eye color gene, it can, I'm not saying ki blue, red, green. I'm saying in this area on the chromosome, if this is a chromosome, in this area on the chromosome, you will find eye color gene. In this area, you will find a gene which gives you extra hair growth. That's it. Okay. So whenever I'm talking about gene, what am I referring to? I'm referring to sequence of nucleotides on a specific place on the chromosome or in the genome. Okay. Now tell me, what is this gene? Yeah. Yeah, that is for your basic level. Next, how will you write an answer? First, it is a functional part of DNA, and it refers to a set of nucleotides situated on your chromosome or in your genome at a particular fixed loci or locus. Okay. Okay. It is a functional part of functional part of genome or DNA. It is a what did I say? Set of nucleotides situated at a particular loci on your chromosome. It is a set of nucleotides situated at a particular loci on your genome or chromosomes or whatever. Okay. So clear? Doubts? Yeah. Now, yeah, that is gene. Okay. Can someone, last class I discussed, right? Why are there so many less genes in general? I told you, if this is a huge area, the construction is only in 3% places. Okay. That too sometimes quite far. Now why is this? Why is this? Not really. Not really. It's like, in general, our DNA tends to mutate. Why? Many reasons. It can be radiation effect, and it can be, radiation can be from sun, can be from your surrounding places, and it can be using junk also. Sometimes mutation happens using when you have a lot of junk food. So, and if you must have noticed, if you smoke, then there's obviously a hair loss. I mean, that is an epigenetic change. It won't change genetically, but it will still change. So lifestyle factors and your environment around will induce some mutations, at least if not now, in the future generation. Okay. So now, in order to imagine, if a mutation occurs, if this is a huge DNA, this is your gene, these are your important gene areas. Okay, let's take only this part. Let's take only Y chromosome, very small chromosome, smallest chromosome, Y chromosome. It has 3 genes. Let's say it has only 3 genes. Now, in this whole Y chromosome, if mutation happens in this part, it's fine, not a big deal. It's not getting coded into a protein. Why? Only these areas are getting coded into protein. That means these areas are getting expressed, right? Only these areas are getting expressed. These areas are not getting expressed. So, if there's a mutation at here or here or here, it's fine, pretty fine. If there's a mutation here, then that's a problem. Why? Let's say for sickle cell anemia or thalassemia. For thalassemia, just in your hemoglobin gene, one part from T changes to A. That's it. You'll have thalassemia. That means your hemoglobin in your body won't be produced properly. Okay. So only one small change can be debilitating. So in general, if you have a big field like this, if you have constructions only here, now imagine there is a pigeon going. Now if it tries to drop its litter, the probability that it will drop on the floor is more than on the construction. Why? Simply going. Small constructions are here and here, but randomly if I throw something, the probability that it will hit the ground is more. That's why we have this junk DNA, because, and that's why we have all the genes not located closely together, far, situated far. Okay. So if there's a mutation in future, it is most likely that that mutation will occur in a part of DNA which is not coding. If there's mutation in future, it is most likely that it will not occur in a part which is on your gene area. Okay, yeah. We are talking about RNA interference technology. We've discussed what is small interference RNA, although I did not tell you technically this is it, but I told you how things work with respect to RNA interference technology. We use a transgenic plant to make something called as, the jargon is important, to make something called as antisense RNA. Antisense RNA. I used a word called complementary RNA because it's more simple. Okay. So it needs to get, so a nematode, a worm, it needs to, it needs its genes to get transcripted into its mRNA, and this mRNA should go to plant's ribosomes. Resource is of a plant, to be translated into protein, food for nematode. Why? This protein is food for nematode. Without this protein, it can't survive. Yeah. Now, how we do it? A transgenic plant is made with nematode specific gene. You have nematode specific gene. Now, this will make your dsRNA. dsRNA is double stranded RNA. And so there is a small change here. There is something called as, I want you to focus here. Jamna, listen. First, this plant which we have made, the transgenic plant, which we've added an extra gene of what? Nematode into a plant, that is called transgenic plant. That will make something called as dsRNA, that is a double stranded RNA. It won't make a single stranded RNA, it will make a double stranded RNA. This double stranded RNA will open up, and the opened part will stick to mRNA of worm. Did you understand? This is a transgenic. This is a transgenic plant. This is the extra gene. This is all the plant gene, extra gene. This will make, I told you, single stranded RNA. No, it will not make a single stranded RNA. It will make a double stranded RNA. This double stranded RNA will go, and this is mRNA of your nematode. Let's say AUGC, nematode. This part will be TACG. This part will open up and get attached. Okay, dsRNA will open up and it complements with the mRNA of nematode. What happens if you complement? What happens? It cannot form into a protein. This mRNA is of nematode, worm. Once the mRNA of the nematode is complemented with other part, it will become double stranded. What will happen when an mRNA is double stranded? It cannot form into a protein. It cannot be translated into a protein. Okay. Should we move to the next part? Because this is going to be an important part, because this is where we discuss Nobel Prize of 2024. Okay, I'll start with a question. Now, as all of us know, all of your body, right from your head, from your hair to all of your, this is a bad representation of body, but yeah, let's say this is good hair. So my point is, all of your cells, even your nails, even your bones, have your nucleus. This nucleus has very same 23 pairs of chromosomes, right? It has 23 pairs of chromosomes, right? When every single cell has 23 pairs of chromosomes, then why is that a particular cell is different from another cell? When the genetic code is same, when all the information is same, then why is it that we have separate skin cells, we have separate hair cells, we have separate liver cells, we have separate nephrons are in kidneys? Why? Did you ever think of it? Because the same genetic material, no? All different cells. And also, right from your birth, you have the very same cell. But only when you hit your puberty, will your body show some changes. Right? Now why? From birth only, you have all the different cells, right? This might sound like a stupid question, but it's pretty natural to question. So this is where you have something called as the expression of a gene. I told you, genes will be expressed into proteins only when they get translated. I repeat, genes will be expressed into proteins only when they get translated. So translation అవ్వకపోతే gene ఉండదు, gene కి use లేదు, right? There is no use for the gene if there is no translation. Now translation can be stopped at many phases. Firstly, if this is a particular gene, now if I stop opening this gene, opening this DNA itself, if I stop opening the DNA, then transcription won't happen. First point. So I'll stop my DNA to be opened. Transcription won't happen. So that particular gene is redundant, or that particular gene doesn't express at all. Now, 1. 2. If it opened, there is mRNA created. mRNA is done, created. This mRNA will now nicely go to ribosomes. It will become a protein. Now, if I stop it at this very particular stage, I interfere, but not using external factors. Let's say my body has some mechanism, I'll tell you what mechanism it is. This stops this mRNA to be getting translated into a protein. That way also my gene expression will be stopped, right? Stopped, right? So these are the two areas. Now, these are the two areas. First I can stop from, and I can also stop at transcription level. Let's say for transcription also, to copy this, I need some elements. They are called transcription factors. For example, when there is RT-PCR test you've taken, what is the full form of the test? Reverse. What? Reverse transcriptase PCR full form. Were you there? Did you listen to the class? Okay, no. Okay, reverse transcriptase polymerase chain reaction. That is the full form, right? I told you 100 times in the class. The point is, reverse transcriptase is an element which you add to your virus. Which virus? You collect swabs from your mouth, and this has which virus? Coronavirus. Coronavirus is an RNA virus. RNA virus ante ikkada RNA untadi kada. Now you have to first reverse transcript into DNA. Then this polymerase chain reaction will make this DNA into many copies, and then it is easy to identify do you have virus or not. So if you add a chemical called reverse transcriptase to RNA, it will change into DNA. If I add transcriptase to DNA, it will change into RNA. If I add transcriptase to DNA, it will change into RNA. So that particular chemical is called transcription factor. Although chemical is not the right word to use, what is the right word to use? Enzyme. Okay, enzyme. Transcription factor is an enzyme which allows you to transcript from your DNA to RNA. Okay, so if I stop the transcription factor, if I stop opening of the DNA, if I stop if I stop after this mRNA is made by using complementary mRNA or antisense mRNA, then I can stop the expression of genes, right? Now, firstly, different, I told you, all cells have same DNA, but why are they different? Why are they different? Hair, skin, muscle. So, firstly, because your gene expression is spatially different, temporally different. Your gene expression spatially in different cells, not every single gene will be expressed. In your muscle cell, only few genes will be expressed. And what is temporally? It is as you grow, few genes will be expressed. For example, when you hit puberty, when for women it's menarche, it's called menarche. So when it happens, then there will be bodily changes. Now, why does it happen at particular age? Why? Because at that particular age, that gene will start expressing itself. So there will be spatial and temporal. Okay, as we grow, puberty, menopause, and that's why there is a theory. Any anthropology optional students? No one? Okay. So there's a theory called death gene theory. That means after a particular age, that gene which is responsible for death will transcript and translate. They say that. That's a theory only. So the point is spatial and temporal. So if someone asks you this question, you should tell me that due to differential gene expression spatially and temporally, our different cells in our body are different. Okay. Why? Despite having the same genetic code, due to differential gene expression. Okay. So now we did not know how did this particular gene expression actually worked. This is where the discovery by these two scientists have revolutionized. That's called microRNA. So scientists, the Nobel laureates have said that it is using microRNA, or rather microRNA is one of the ways to stop the gene expression. They have explained how this microRNA actually changes your gene expression. How your microRNA changes your gene expression. That is the Nobel Prize. Okay. So microRNA stops gene expression spatially and temporally. So we'll talk about applications. First I'll tell you how does it stop. Okay, first we'll read everything which is here, then it will make more sense, because see, these notes are needed for UPSC, and what I'm explaining is needed for understanding. Okay, so without understanding you can't remember something. So firstly, what is coding part of DNA? Coding part of DNA, genes. You can't, let's say for all the practical purposes and for simplicity, we'll call coding part of DNA as a gene. Okay, although it is not the definition. Produces mRNA. We all know this. Genes produce mRNA, mRNA produces protein, okay. Yeah, focus in the class, okay. Non-coding part of DNA. This is junk DNA, right? Again, for all simplicity, it's called junk DNA. Now, this first fundamental difference, non-coding part produces microRNA. Now you might ask, sir, this is a misnomer, because this is also getting coded. In some way it's getting coded, no. So non-coding, coding is for us to understand. Okay. So genes produce mRNA, non-coding part, that is non-genes, produce microRNA. First fundamental difference. What is if someone asks you what is a microRNA, you have to tell them that the RNA which is produced by the areas which are not gene areas. Okay, the areas which are not gene areas. Right? The RNA which is produced by the areas which are not gene areas is your microRNA. Now, microRNA is typically small, 19 to 22 strands, okay, and it is single stranded. First difference, I told you, from areas which are non-DNA, so non-genes. Now, second difference is that very small. mRNA is a little huge. MicroRNA is very small. Okay, it is single stranded, and microRNA is partially complementary to messenger RNA. I use the word partially. What is partially? What is partially? Only a small part, right? So now you see, this is microRNA, this is mRNA. Now, not all of the microRNA is complementary with your mRNA, right? Only a small part. Excuse me. I don't want anyone to hold hands like this in my class, okay? This is a closed position. Open your hands and listen. Yeah. So I don't want. Yeah. Sorry. MicroRNA doesn't what? Sync with mRNA at all the places. MicroRNA only syncs at a particular place. That's why I called it partially complementary. So it works something like this. Okay. Now, what happens if I complement messenger RNA? What happens if I complement it? Yes, protein will not be formed. So this mRNA will not be expressed. Will not be translated into a protein. What will happen instead? It will somehow break down and get digested in your cell itself. MicroRNA is now stopping your messenger RNA. MicroRNA is now stopping your messenger RNA. That means, can I tell you that microRNA is actually stopping some of your genes to be translated into proteins. Now, can I tell you that in younger ages, there will be few genes which will not be translated. That can be due to microRNA, right? That can be due to microRNA. Why? Because microRNA, after your gene is getting expressed into mRNA, is stopping mRNA from getting translated into a protein. Did you understand this? Cool? Any doubts as of now? You? Okay, so next, this mRNA, this messenger RNA now cannot be translated as it became double-stranded. This floats as mRNA, which is unfit for translation, useless, broken down and digested within the cell. Alright? Yeah. Now, you see, why is this really important? Because they've actually told you that there is a mechanism in your body which is stopping your genes to be expressed, and that too from your junk protein. So they have opened a new world. Now, can you, if I make this microRNA in lab, which I can make polypeptide synthesis. If I make this microRNA in the lab, and if a particular person is having a sickle cell anemia, what is sickle cell anemia? Did we discuss in the class? We did discuss, right? My first class, sickle cell anemia. So when your RBCs are typically biconcave. But in sickle cell case, what happens? It is in a sickle. Sorry, what is a sickle? Sickle is this Communist Party's symbol. Okay, my skills of diagram are bad, but this is your sickle. Okay, so your RBC, which is supposed to be like this, smooth, is now like a sickle, right? So what happens if there is a sickle? If there is a lot of sickling, if this is your vein in your blood vessels, now your RBCs will easily flow, which healthy RBCs. But if you have sickled RBCs, they'll get stuck here. They'll obstruct the flow. Now what happens with the sickle cell people? I told you they cannot run a lot because their hemoglobin capacity in this is less. Yes. Also, these healthy RBCs live up to 120 days. These die in 20 to 25 days max. Okay. So if their blood keeps on dying, what happens? They can't produce. They can't do a lot of exercise, right? They're generally weak. Their immune system is a little problematic. So now, what in the sickle cell anemia, what happens is that there's a particular gene in your bone marrow. This gene changes into mRNA. This mRNA will make this sickle cell. I mean, this RBC is made by your gene only. It's telling to make like this. It's making. So the recipe is itself faulty. Now, for you to change a person's recipe, if it is not at embryo level, there's only one cell. No? Embryo, one cell, hardly few cells. Zygotic level also, hardly few cells. That you can count these cells. In this cell, it is easy to change, from nucleus, it is easy to change all the 23 pairs of chromosomes. It's easy. If there's only one cell, you can take its nucleus, add a gene, remove a gene, do whatever, put it back. It might work. But now a person is grown. Every single cell of his has what? These 23 chromosomes and a defective gene. Every single cell has. Especially in your bone marrow, there are millions of cells. Now there have to be bone marrow transplant cheyali. And even if you find first of all, finding a right donor is hard. Even if you find the right donor, your body's immune system will only attack. So it is very hard. They have to live on immunosuppressants. That means they have to, even if there is a small cold for the people who got bone marrow transplant, they might die because their immune system is being suppressed artificially by medicine. Why? Because if you don't do it, this bone marrow will be attacked by your own body cells. Autoimmune disorder, they'll die. So this is a very hard task. If you have to do bone marrow transplant also, and that is also risky. Not everyone can do it. And okay, let's say your parents are smart. They've stored your umbilical cord while you're giving birth itself. Now using that stem cells from your umbilical cord, you have to develop your own bone marrow, which can be transplanted. Then this is also a complicated procedure and a very costly procedure, right? Now, what if I tell you that fine, your recipe is disabled. I mean, your recipe is bad. Fine, but I'll not let your recipe, that is your mRNA, actually translate into a sickle cell. I'll not do that. I'll stop it here itself. Although it's chala theoretical only, I'm trying to tell you which is not practically possible. There are two different CRISPR-Cas9 treatments for sickle cell anemia which have been approved very recently. We'll discuss in the class. But this is just a theory, okay? Just understand it as a theory. Don't take it completely as truth. So if I tell you that this particular mRNA level, I can stop this gene expression. I can stop from your, I can stop making sickle cells. Okay. Now, how will I do it? I'll artificially give him every week or every day microRNA, which is made in lab. What is this microRNA? This microRNA, which can stop this mRNA. What if I give him? That's great, right? So sometimes directly into the bone marrow, or even if not directly into bone marrow, somewhere, let's say, see, this bone marrow example is something which I made up, okay? This is not actually correct. What happens is, you can't do anything, if there's this sickle cell anemia thing. So I'll tell you what exactly is done with sickle cell anemia. But imagine there's another thing altogether. So with this microRNA, I can actually fight the genetic diseases. That is something you have to know. Okay, UPSC will ask you only one thing. Using this microRNA technology, can you fight genetic diseases? Yes, you can fight genetic diseases. Now, this is the application part. I can fight genetic disorders. Why? Because we can make miRNA in the lab. And what is this miRNA? Anyone? What is this miRNA? I told you, RNA which is made from your non-gene areas, which is partially complementary to your messenger RNA. RNA which is made from non-gene areas, which is partially complementary to your messenger RNA. Now if I can make this microRNA, that will now complement my bad mRNA, which I don't want it to be translated into protein. I can fight genetic disorders. I can fight Alzheimer's. Alzheimer's is something like dementia. You forget, right? Actually, forgetting is not the right word. Degeneration of your neurons in your brain. That is the right word. Cancer research, you can use it. Then gene silencing. Gene silencing. I told you there is a specific term called gene silencing. You are silencing the gene without changing the gene, right? Using microRNA. Okay. Yeah, that's it, folks. This is the Nobel Prize, and this is something how this actually works. This is your DNA. DNA now opens up, denaturation. This DNA becomes mRNA, microRNA, and this microRNA is partially complementary to. This microRNA is partially complementary to messenger RNA. Okay, this is messenger RNA. This is microRNA. Messenger RNA changes into protein, but when this messenger RNA is confronted with microRNA, this does not change into or translate into a protein. Now, what are the issues? Whenever there is a thing, there will be issues. Toxicity is a problem. What if you have designed this microRNA for a specific purpose, but as often as we say in science, the most complicated system is your human body. You don't even know completely about it. Now you designed this microRNA for a specific purpose. Now there's something else which is happening with this microRNA. This microRNA is not getting, this is your target messenger RNA. You want to target this. Now this part is targeting this. What if this particular part is targeting some other messenger RNA and your body is getting affected? Right, that's a problem. Toxicity is a problem. Autoimmune disorders. What if your own body cell now changes and attacks your own body cells? Why? Because it's seeing a lot of RNA around. That is also a possibility. Although I don't know how exactly this happens, but you don't need it. You simply have to know what is autoimmune disorder. The examples of autoimmune disorders: rheumatoid arthritis, autoimmune disorder, things like that. Okay.